Okay, let's just jump right in. Forget everything you thought you knew about how cells in your body talk to each other. It's not always, direct contact or simple signals.
No, it's much more complex. Turns out they're constantly sending out these tiny packages. Think of them like biological mail carriers.
Little messengers. Filled with what
Filled
exactly?
with complex instructions, molecules. They send them out into the space between cells. These are extracellular vesicles, EVs.
EVs. Okay, and they're basically bubbles of fat, lipids, right? Secreted by cells.
Exactly, lipid-bound vesicles. And inside they carry all sorts of bioactive stuff, proteins, nucleic acids, metabolites, lipids. It's how cells transfer real information, actual material.
So they cause actual physiological changes in the cells that pick them up.
Absolutely. It's not just a quick note. It's more like sending a whole toolkit or a set of instructions.
Yeah, okay. And the contents, the cargo, as you call it, that varies.
Oh, definitely. It depends hugely on the cell sending it. Its tissue type, its immune status, what's going on with it at that moment,
Mhm.
the cellular context.
So it's a very targeted message system.
Highly specific.
Mhm.
And that specificity, that capability, that's what makes them so incredibly interesting and important, potentially.
Which is why we're doing this deep dive today, isn't it? Into this whole world of EVs, using the insights from the sources you shared. And we're putting a particular spotlight on one type the sources really seem to zoom in on, exosomes.
Yes, exosomes are definitely a major focus in the research right now and in these sources. They do mention other types like microvesicles and apoptotic bodies, though one source uses the term autophagosomes a bit differently.
Right, we'll touch on those. But exosomes seem to be where a lot of the detailed action is described.
Absolutely. Understanding them has really shifted how we think about cell communication. It's been, well, transformative.
So let's nail down exosomes first. What makes an exosome specifically?
Okay, so their defining feature is their origin. They actually start their life inside the cell as even smaller vesicles floating within a larger compartment.
Inside, not just pinching off the outside.
No, that's key. They are technically called intraluminal vesicles, ILVs. Intraluminal meaning inside the lumen or space of something else.
And what are they inside of?
They're found inside structures called multivesicular bodies, or MVBs. These MVBs contain many of these little ILVs.
Multivesicular. So multiple vesicles inside one body. Okay, that makes sense. How does that form?
It's this really fascinating process called the endosomal pathway. It starts with the outer cell membrane budding inward. That's endocytosis. It creates an initial bubble inside called an early endosome.
Okay, so the cell takes something in from the outside, forms this early endosome.
Right, but then, and this is the cool part, the membrane of that endosome itself starts budding inward. Again,
Whoa, wait. So the membrane of the thing that just budded inward now buds inward again.
exactly, like membrane inception, kind of. This inward budding of the endosome's own membrane creates those tiny ILVs, the future exosomes, floating inside the maturing endosome.
And that structure, full of tiny vesicles, that's the MVB.
That's the multivesicular body. And these MVBs, they're not just holding chambers, they're like crucial sorting stations inside the cell, involved in trafficking proteins and other materials around.
So the cell is actively packing specific cargo into those little ILVs as they form within the MVB.
Precisely. It's a sorting and packaging process.
Yeah.
And the sources mentioned the machinery involved, right? The ESCRT pathway.
Yes, ESCRT. Endosomal sorting complex required for transport. That sounds important.
It is. It's a key set of proteins that helps drive that inward budding process, forming the ILVs and sorting cargo into them. It's complex molecular machinery at work.
Okay, so now we have this MVB, packed with future exosomes. What happens to it? It doesn't just sit there, does it?
No, it has two main potential fates. One route is fusion with lysosomes.
The cell's recycling centers, basically, break stuff down.
Exactly. If the MVB fuses with a lysosome, all those exosomes inside and their cargo get degraded, destroyed.
A dead end for those messages. What's the other path?
The other path is secretion. The MVB travels to the cell's outer edge, the plasma membrane, and fuses with it.
So the MVB membrane merges with the cell surface membrane.
Right. And when that happens, the MVB essentially turns inside out, releasing all those little ILVs, which we now call exosomes, out into the extracellular space, outside the cell.
And they're free to travel to other cells. Now, one source had this really specific, intriguing detail about what decides which path the MVB takes, lysosome or release.
Yeah, that was fascinating. While it's still being fully worked out, the research pointed to the amount of cholesterol in the MVB's own membrane.
Cholesterol. Really?
Seems so. The idea is that MVBs rich in cholesterol are preferentially directed towards the plasma membrane for release. Those lacking sufficient cholesterol are more likely targeted to the lysosome for degradation.
Wow. The fat content of the vesicle membrane directs its fate. That's
Yeah.
incredibly precise cellular logistics.
It really highlights the complexity, doesn't it?
Yeah.
And, it's funny, when scientists first observed these
They know
vesicles,
what they're looking at.
well, they saw them, but they thought they were just cellular trash bags. A way for cells to get rid of unwanted proteins and debris. Cellular dustbins, essentially.
Seriously, just waste removal.
That was the initial thought. It shows how far our understanding has come. We now know they're absolutely vital communication tools involved in maintaining balance, homeostasis, and unfortunately also in disease processes.
And they're not just stuck in one place. The sources listed a huge range of body fluids where you can find them.
Oh, they're everywhere. Plasma, serum, urine, cerebrospinal fluid, the fluid around your brain and spine, breast milk, saliva, tears, amniotic fluid, lymph, even stomach acid. Pretty much any body fluid you can think of has exosomes in it.
That accessibility is key, isn't it? We'll definitely come back to that when we talk about medical uses. But first, let's quickly touch on the other EV types, just for comparison. Microvesicles. How are they different?
Microvesicles are different mainly in how they form. They don't come from inside via MVBs. Instead, they form by direct outward budding or blebbing from the cell's main surface membrane, the plasma membrane.
So they just pinch off directly from the outside, like a little bubble.
Exactly like that. And they tend to be larger than exosomes.
How much larger?
Exosomes are typically in the range of, say, 30 to 150 nanometers. Really small. Microvesicles are usually bigger, maybe 100 nanometers, up to 1 micrometer, which is 1,000 nanometers. So potentially quite a bit larger.
Okay, different origin, different size. What about how they're made?
That process is maybe less clearly understood than exosome biogenesis via the ESCRT pathway. But we know it involves the cell's internal skeleton, the cytoskeleton and molecular motors, things that push and pull, plus machinery for membrane fusion.
And the production is sensitive to the cell's condition?
Very much so. Stress, activation state,
Right.
it all affects microvesicle release. And interestingly, for a recipient cell to take up a microvesicle, it seems to require energy. It's an active process for the receiving cell.
Okay. And potential uses are similar to exosomes.
Broadly speaking, yes. The clinical applications being explored often overlap biomarkers, maybe therapeutics.
Right. Then there was that third category, apoptotic bodies or autophagozones, depending on the source. What's the deal there?
Yeah, a slight difference in classification. One source talks about apoptotic bodies. These come from cells that are dying undergoing programmed cell death, or apoptosis.
So breakdown products.
Kind of. As the cell dies, its membrane pulls away from the internal skeleton, and it breaks up into these membrane-bound chunks, which can be quite large, 50 up to 5,000 nanometers. They contain bits of the dying cell, organelles, DNA fragments.
Okay, released during cell death. And the other source mentioned autophagozomes.
Right. Autophagozomes are usually involved in the cell's internal recycling process, autophagy breaking down old or damaged parts. This source suggested a pathway where these might sometimes be released outside the cell, perhaps by fusing with MVBs or the plasma membrane instead of just the lysosome.
So it may be a non-classical release related to internal cleanup. The main point seems to be they're distinct from the active intentional secretion of exosomes and microvesicles from generally healthy cells.
Exactly. Different origins, different signals, perhaps.
Mm-hmm.
It paints a picture of a whole ecosystem of different vesicles being released for various reasons.
It's definitely complex. Okay, let's get back to the functions, especially for exosomes. We said communication, homeostasis. What else?
Well, a huge area is the immune system. Exosomes are critical players there. They can carry molecules, antigens from their parent cell and present them to immune cells, basically activating an immune response.
Like little messengers alerting the immune system?
Precisely. Saying, hey, look what's going on in here. And their roles can be very specific to tissues. Like in the nervous system, the sources mention exosomes helping make myelin, that nerve insulation.
Which is vital for nerve function.
Absolutely. And supporting neuron survival, growth, promoting repair after injury. All sounds incredibly positive.
It does. But
Yeah.
there was a big but in the sources, wasn't there? Especially regarding the brain.
Yes. This is the dark side, you could say. The very same exosome system that supports nerve health can also, unfortunately, be involved in spreading harmful proteins.
Pathogenic proteins.
Exactly. Proteins linked to neurodegenerative diseases. The sources specifically name-drop beta-amyloid peptides, which you find in Alzheimer's plaques, alpha-synuclein, linked to Parkinson's, and superoxide dismutase, relevant to ALS.
Wait, hang on. The cell's own delivery system can spread the very things that cause these devastating diseases.
That seems to be the case, yes. It's a deeply concerning finding highlighted in the sources. It means these natural communication pathways can be hijacked or simply contribute inadvertently to disease progression by transporting these misfolded, dangerous proteins between cells.
That's quite unsettling, the mail service spreading the poison.
It's a stark example of their dual nature. Essential for health, but potentially complicit in disease.
What about other functions mentioned? General EV roles.
The sources also mention EVs initiating immune responses against pathogens, like viruses or bacteria, and even against tumors. They help maintain that internal balance, homeostasis, partly by getting rid of potentially harmful stuff, like bits of DNA that might trigger inflammation if they stay inside.
And involvement in big processes.
Yeah, things like pregnancy, stem cell differentiation, guiding cells to become specific types, and coordinating recovery from injury.
There was a specific mention of mesenchymal stem cell EVs, right, from fat tissue.
Yes, adipose-derived MSCs. Their exosomes seem packed with goodies that promote cell growth, migration, new blood vessel formation, angiogenesis, nerve regeneration, and modulating immune responses. Real therapeutic potential there.
Sounds like a potent repair kit in a tiny package. But again, the flip side, cancer.
Right. Exosomes can also contribute to cancer progression. They might carry growth factors that stimulate tumor cells, promote angiogenesis to feed the tumor, or carry molecules that suppress cell death or trigger inflammation, all helping the cancer grow and spread.
So, involved in this incredibly complex network, heroes and villains, depending on the context and the cargo, given all this and how accessible they are in fluids, the medical potential feels enormous.
It truly is, and the sources really emphasize this. A major area is using them as biomarkers.
For diagnosis.
Exactly, because they're in blood, urine, saliva. You can get at them easily. No need for invasive biopsies in many cases. It's the concept of a liquid biopsy.
Ah, liquid biopsy. Analyzing fluids to see what's going on deeper inside the body.
Precisely. And exosomes are ideal carriers for this, because their cargo is like a snapshot of the cell they came from. If a cell is cancerous or stressed or infected, its exosomes will reflect that.
And the sources gave concrete examples.
They did. Promising research on detecting alpha-synuclein in plasma for Parkinson's diagnosis, finding markers for brain tumors like glioblastoma, using urine exosomes to spot acute kidney injury, identifying potential markers for pancreatic and lung cancer.
That's huge. Early detection for some really tough diseases.
The potential is definitely there. The sources indicate some real successes in these research areas, suggesting they could become powerful diagnostic tools.
Okay, diagnosis is one thing. What about actually treating diseases with them?
That's the other big frontier, therapeutic applications. First, you can track exosome profiles to monitor how a patient is responding to treatment. Are the bad signals going down? Are good signals appearing?
Makes sense. What else?
Vaccine development is another area. Because they can present antigens naturally and are well tolerated by the body, they might be useful platforms for creating vaccines. They stick around for a decent amount of time, too.
Like using the body's own messenger system to train the immune system.
That's the idea.
Yeah.
But maybe the most talked about potential is using them as drug delivery systems.
Ah, loading them up with medicine.
Exactly. Think about it. They're naturally designed to travel between cells, cross membranes, and deliver contents. So why not hijack them? Load them up with therapeutic cargo RNA, proteins, small molecule drugs, genetic material.
And send them to specific targets, like cancer cells or damaged tissue.
That's the goal. The sources mention this potential, especially using exosomes from, say, mesenchymal stem cells, which already have regenerative properties, perhaps even as therapeutic agents themselves to reduce tissue injury.
So the vesicle itself could be the therapy, or it could carry a drug payload.
Both are being explored. Of course, the techniques for efficiently loading specific cargo and targeting the exosomes precisely where you want them to go, that's still under heavy development, but the natural advantages are clear.
Using the body's own nano delivery trucks, it feels elegant. Now, there was that other really wild connection mentioned in the sources, particularly the second one, between EVs and viruses.
Ah, yes, that's a fascinating intersection. It turns out that the cellular machinery cells used to make, package, and release EVs. Viruses often use that exact same machinery for their own purposes.
Viruses hijack the EV production line.
They absolutely can. For their own replication, assembly, and crucially, for getting out of the cell and spreading. Some viruses can even ramp up EV production in the infected cell.
Wow. And they can put their own stuff inside the EVs?
Yes, they can manipulate the process so that EVs carry viral genetic material or viral proteins, essentially using the cell's own communication system to spread the infection.
That is clever and quite scary.
Yeah.
But it wasn't all bad news, was it? EVs can fight back.
Right, it's a double-edged sword. Infected cells can also package viral components or immune-stimulating molecules into EVs, specifically to warn neighboring cells or alert the wider immune system.
So the EV becomes an alarm signal. Warning, virus detected.
Exactly.
Yeah.
So EVs released during a viral infection can be incredibly immunomodulatory. Depending on the cargo, they might stimulate the immune system, which could be good, but might also contribute to damaging inflammation if it goes on too long.
Yeah.
Or they might inhibit the immune system. This could help the virus hide, evade immune clearance, and potentially even contribute to cancer progression if the virus is oncogenic.
So it really depends on the specific messages packed inside those virus-related EVs.
It really does. And understanding this interplay, this crosstalk between viruses and the EV system, is opening up some really exciting ideas for immunotherapy. This applies to both RNA and DNA viruses, by the way.
And that led to that really futuristic-sounding cancer therapy idea.
Yes, still very much developmental, but incredibly promising. The idea of packaging oncolytic viruses inside EVs.
Oncolytic viruses, those are the viruses engineered to attack cancer cells, right?
Correct. So the idea is to put these therapeutic viruses inside a natural delivery vehicle, an EV.
Why? What's the advantage?
Several potential advantages. The EV might act like a cloak, shielding the virus from the patient's immune system so it can reach the tumor intact. It might also improve how well the tumor cells actually take up the virus.
So better targeting and protection for the virus payload?
Potentially, yes, enhancing the effectiveness of the viral therapy. It's a really smart strategy, leveraging the body's natural systems for a therapeutic purpose.
It really is. Okay, let's try and wrap this up. We've gone from realizing cells send out these tiny packages
Vesicles, once dismissed as mere cellular dustbins.
to understanding they're fundamental messengers, crucial for health but also involved in disease. We focus particularly on exosomes and their unique origin story, forming inside the cell within those MVBs.
Right, through that intricate endosomal pathway, with their release possibly even tuned by things like cholesterol levels. And we saw how they differ from microvesicles budding off the surface, or apoptotic bodies from dying cells.
And crucially, they're everywhere in almost all our body fluids. That makes them accessible.
Incredibly accessible.
Yeah.
Which leads directly to that huge clinical potential. First, as biomarkers via liquid biopsy. Imagine getting earlier warnings for Parkinson's, brain tumors, kidney injury, cancers, just from a blood or urine test.
That's transformative potential right there. And then the therapeutic side, using them as natural drug delivery systems.
Loading them with cargo, maybe even using stem cell exosomes directly for tissue repair, and leveraging their immune roles potentially in vaccines.
And finally, that complex dance with viruses. How viruses hijack the system, but how cells can use it to fight back. And how we might even use EVs to deliver cancer-fighting viruses more effectively.
Such a dynamic field. Honestly, understanding these tiny vesicles is key to unlocking so many potential breakthroughs in diagnosis and treatment across a huge range of diseases. The impact on your future health could be immense.
It really could. So maybe a final thought for everyone listening, something to chew on from these sources. Think about this hidden world, this constant chatter happening between your cells via these exosomes. They're involved in the everyday running of things and fighting infections, but also potentially spreading diseases like Alzheimer's or cancer. And now we're looking at harnessing them, using them as therapies. How much more is going on down there at that tiny scale that we just haven't uncovered yet? It really makes you wonder, doesn't it? These minuscule structures holding such massive potential for good and ill. What else are we missing? Thanks for taking us through these sources today.
My pleasure. It's a truly fascinating area.